From Microbe TV, this is TWiP, This Week in Parasitism, episode 153, recorded on June 12th, 2018. I'm Vincent Racaniello, and joining me today, right here in New York City, Daniel Griffin.
Hello, everybody.
Where the waters outside the window are choppy, right?
There's a breeze is up. There's a little bit of some white caps on the river. It looks beautiful out there.
So the white caps usually come from the breeze, right?
And it's at a certain breeze. Once it gets up to, let's say, about 15 knots of breeze, about 15 miles per hour roughly, you start to see the white caps, and that's when the sailing gets more exciting.
So you could tell roughly how strong the wind is, although it could be more than 15. You would still have white caps, right?
You actually start, and the more it goes up, the more white caps you see. So you start to see a few, and then it increases.
So do you have a calibration in you that would automatically say that's 20 knots, that's 30 knots?
I feel like my dad is better at this than I am.
Right. Okay.
I think people who are more senior and more experienced probably even better at this.
Well, Dixon is off somewhere. We're not sure where in his travels. He's been around. He's been away for a while, and so have I. But I have a feeling he was going to Shanghai.
Mm-hmm. I do think I remember. I remember he was talking about how they hand him envelopes full of cash, and that's why he goes.
Right. So this is about the second episode Daniel and I have done together. So we hope you don't turn it off right now, right, Daniel? Continue.
Well, we don't have to worry about, we've already picked on Dixon in his absence. So
that's right.
I was going to say we don't have to worry about not being nice to Dixon, but we're already. No, I'm sure he's over there spreading the word about alternative farming techniques, helping to feed the world through new methodologies, which is a majorly important thing.
Sure. I was in the Netherlands last week, and to and from the airport, you go through fields and fields. It's flat. It's the lowlands, right? It's all farm. It's all outdoor farming. I guess they have enough land, but they haven't done the indoor stuff yet.
It's interesting. They've worked pretty hard to get that land. So
yeah. And I'm sure Dixon would have a few words to say about that, but he can't. So we will move onwards. Now, the first thing we have to do here is we have a follow-up from David P., who we picked last time as the winner of the book. And he writes, "Dear hosts, I am happy that the RNG gods," which is random number generator.
I had to ask what that was.
"Smiled upon me yet again to win a signed copy of Parasitic Diseases. I would love to give the copy to a faculty member, but I would also not mind yielding the copy to another individual who guessed in episode 152. For the sake of fairness, I would prefer to let the book go to another TWiP guesser." What a nice person we have there, right?
I agree. It speaks highly for David P., his generosity.
All right, so I do random number generator. Push the button, and we have number 8. Now, an 8 is an email that I just added, actually.
But are we picking from episode 152?
Yeah.
Oh, okay.
This is one we didn't read, but it came in afterwards.
Should we read it?
Go ahead.
Sue Ellen. Oh, okay.
Is that how you pronounce it, Sue Ellen?
Sue Ellen. We were corrected, and then, Dixon, because he's not here, will continue to harass him. I kept pronouncing it Swellen. But let us get to, and they're numbered. You've really done a good job here. Sue Ellen wrote, "The case of the woman in Panama who has abdominal bloating and greasy stool, who was apparently wrongly diagnosed with an ovarian cyst. Once again, I was really happy to be able to come up with at least one possible diagnosis just by hearing the symptoms without having to look anything up. Geodiasis, from the protozoa that can be found all over the world in water sources. A good description of the symptoms can be found on page 15 of Parasitic Diseases, 6th edition. The acute diarrhea of Geodiasis is classically described as foul smelling with flatulence, nausea, weight loss, and abdominal cramps with bloating. It does appear that a positive diagnosis requires a microscopic examination of the stool, which I'm guessing is not possible so far out in the wilds of Panama. So perhaps it would be enough to treat the patient with metronidazole and see if that clears things up. Another possible suspect for this case would be Cryptosporidium, although this usually produces vomiting and nausea in addition to diarrhea. Amoeba Histolytica also causes diarrhea, really, so many parasites do. But again, I don't think it's as good a candidate for this diagnosis as Giardia. Hope I got this one right, and hoping I can win an autographed copy of PD6. And
here you go, Sue Ellen. And just send me your address, TWiP@microbe.tv. Okay, next follow-up came from Kathy Spindler from TWiV, who wrote to say that peeps means people. Last time we didn't know what it meant, remember?
We're not sure. I thought it was a reference to one of the parasitology fiction novels.
No, last time we didn't know what peeps was, you and me and Dixon. And in fact, that was the name of I gave that the episode name.
Oh, really?
Of our last...
now we actually know what we were discussing.
Here we go. The name was actually A Range of Rads, but I described it as the TWiP peeps solved the case of. So now we know that peeps means people.
Okay.
All right. And that brings us to our case from last time. Daniel, refresh us.
Yes. For those clicking back in and for those clicking in the first time, we presented a case of a 40-year-old man, and this was in the same remote Panamanian village that we had described before, accessible only by several hours in a boat. And this unfortunate gentleman had diarrhea for several weeks. He had noticed blood mixed in with the stool. We got a little history of him working in the fields. No weight change, no fever, felt very tired. He had the exposures of drinking water from the rooftop, living in a home with wooden slat floors with a little gap so things fell between them. No electricity, many animals. No one else was sick. He uses an over-the-water toilet, or he goes just out in the fields. He has lower belly tenderness. He had a normal rectal exam, and we summed it up by saying he was a farmer with 3 weeks of bloody diarrhea.
All right, we have a couple of guesses. One is from Philip. Dear TWiP professors, thank you for taking the time to broadcast these TWiP episodes. They have kept me intellectually charged as I study for board exams. So that's what you take after medical school, Daniel? Is that called a board exam?
Once you've finished, you've got your degree and you go into any particular specialty, there are boards for each specialty. So family practice, internal medicine, and then even the subspecialties, infectious disease.
So you have to do a residency first to take a board exam?
You do.
Okay. Got it.
Then you are, as they say, board eligible, and then you have to pass the exam. And some of the exams, I remember, at least when I took internal medicine, it was 30% failed. And so here, it was really disheartening. So here are people that they've finished medical school, they've finished a residency, they've passed all the exams, and now they're ready to go out. And yet a third of them fail and have to study up and retake the exam. So they're pretty rigorous. And now they have instituted, and I barely missed this grandfathering in, where every 8 or every 10 years, you either take an exam or you do yearly continuing education to maintain your current board status.
So these exams are based on, is it book knowledge, practical knowledge, or both?
It's supposed to be a combination of both, but it ends up being a lot of book knowledge.
So you have to study.
You have to study. I mean, they'll come up, what's the current best treatment for multiple myeloma? And the next treatment will be, what is the most common side effect with that medication? And so there, a lot of the stuff that, we hem and haw we would be looking up, but we cram it back in for the boards to keep ourselves current.
It seems to me if you're interested in that area, you wouldn't mind studying again, right?
I enjoy it, right? I'm sort of a lifelong learner, a lifelong test taker. So.
I mean, I'm sure a lot of people are just charging to get out there and start working, right? But all right, Philip continues. For the case study on TWiP 152, I would be sure to rule out an intestinal Amoeba Histolytica infection. Keep up the good work. Be nice to Dixon. Philip is from Long Island, New York. I wonder if he goes to Hofstra.
I wonder where Philip. Is
many
from.
Medical schools on Long
Island. Well, I guess
Island,
if he's studying for his board exams, I wonder where he is in his training.
Well, you would have either...
Right, he probably would have finished, right, his residency.
Right. So residency typically done at an academic hospital, or it could be a non-academic?
It could be a non-academic, yeah.
Okay. So he could be at any number of hospitals on Long Island, right?
Exactly.
Now, I would think that taking the TWiP cases would be like studying for a board exam, right?
So it's actually, I'll do this as an interesting comment. I recently have started assisting with the University of Glasgow program, which I will see will come up again, I guess. Their global health program, and they actually have a link where people in their global health program have a link to TWiP.
Cool.
And so they'll listen to TWiP, or they'll go to the Parasites Without Borders and they'll watch those lectures. So it's nice to see.
It's a different way of studying, and mixes things up, makes it more interesting. And I would think solving a case would keep, like he says, keep you sharp, right?
It does. And I think it's hard. A lot of people in academics, right? There's a lot of demands in academics. Maybe you're a PhD, you're doing bench research. It's hard to see the number of cases. And it's always, I find it much more memorable when you see a case of something to then put all the knowledge together.
Totally, right.
So hopefully we're vicariously giving them that experience.
As opposed to saying, what is the best treatment for Geodiasis, right? Yeah. I mean, that's, well, give them a case. And
then you remember, what did we give her and why?
Exactly.
Exactly. And how did she tolerate it? And did she agree not to drink beer for a week? Or,
Right. All right. Take the next one, please.
Alexander writes, for a warning, I don't have any background in medicine, but I'd still like to try. Hi, all. I'm a recent graduate with an Associates of Applied Science and Natural Resources as a forestry technician. However, during my education, I also took a liking to oceanography and phytoplankton taxonomy. I started listening to this podcast from NPR and grew to like it a lot. So I guess here's my differential.
Oh, hold on. This is not an NPR podcast, is it?
Not that I'm aware of.
We would like it to be.
Do you think this is when we were ridiculed on NPR?
Must be that was that the contest?
It was. They had the contest where they would ask them, "Is this a real podcast or not?" And they read our little tagline.
I'll bet that is.
And they said, no, that can't possibly. Nobody would want to listen to a podcast about eukaryotic parasites every week.
I think it must be. And, it's interesting. This is not the first person who has come over from that radio show, which shows you the reach of NPR, right?
It's both Dixon and I were listening to that show when we were mentioned. So
many people have heard it. And those of you who are here because of it, please stay.
We'll try to keep you. With the possibility for waterborne pathogens, I got led into looking at Giardia lamblia, Cryptosporidium parvum, Amoeba Histolytica, and Balantidium coli. For G. lamblia, it is waterborne, but the symptoms don't really add up. For C. parvum, I think I want to rule it out for the weight loss and watery diarrhea. However, you did mention in your previous episode that you can still eat more to balance out the weight loss. E. histolytica causes dysentery and can be fatal if left untreated. Also, most exposed individuals are asymptomatic. B. coli has similar symptoms to E. histolytica, but it has also described as lack of energy. Given what I think I know, which is not much, I would say that it could be E. histolytica or B. coli. I would say to prescribe metronidazole, as it is listed as an effective drug for most of what I mentioned other than C. parvum. P.S. The books that I used to help identify phytoplankton differed from the parasitic diseases text in amount of photos and information given on specific entities or entries. Mine would emphasize shapes and just have a little blurb, while parasitic diseases would have long blocks of text that really went into a lot of detail, most of which I don't even comprehend.
Eric writes, hi, trypanosomes. I don't write in often, but I have a hunch about the diagnosis on this week's case. The symptoms described seem pretty straightforward. There aren't a huge number of pathogens that cause dysentery, as far as I know, so that narrows down my guesses to just a few. If I include bacterial infections in my differential, as Dr. Griffin recommended, my list would be the following: Amoeba histolytica, Shigella, Salmonella enterica, and Trichoriosis trichiura. I would rule out the 2 bacterial pathogens, not just because this isn't an episode of TWiM, but because both shigellosis and salmonellosis can cause quite severe symptoms that would be immediately noticeable and distressing, but this patient described not feeling too terribly sick, just tired with some abdominal pain and blood in stool. That leaves two potential diagnoses from my list, E. histolytica and trichoriosis. E. histolytica is well known to cause bloody, watery diarrhea and can also cause fatigue and weight loss, and symptoms can last anywhere from a few days to years. Trichuris trichiura, or whipworm, can be asymptomatic if the person is infected with a low worm burden, but heavy worm burden can result in signs and symptoms that include abdominal pain, bloody or mucousy diarrhea, and even anemia in severe cases, which may cause tiredness. I have to admit I'm having trouble deciding which of those two pathogens is most likely. Both are present in Central America and can infect adults, causing the symptoms described, and both infest the large intestine. Dr. Griffin said that the rectal exam appeared normal. I'm not sure if that would include looking for worms. I would guess not, right? A worm wouldn't be a normal.
It's kind of hard actually on the rectal. I mean, if you maybe there were female adult pinworms, you might see them around the anus.
That's right.
But you're usually not going to visualize, even if a person has an ascaris, right, a pretty large worm, you're not going to usually see it when you do the rectal.
Yeah. Probably the best way to determine which of these two pathogens was the culprit, if it even is one of these two, would be to do a stool O&P and look for distinguishing eggs or larvae or cysts. Would that kind of test be feasible in the rural, low-resource environment described? My hunch is that the man has trichoriosis rather than E. histolytica, though, just because of the localization of the pain and the fact that the stools are not described as being excessively watery. Treatment would be mebendazole, whereas for trichoris, whereas for E. histolytica it would be metronidazole. Maybe both drugs could be given at once. I don't know if that would be safe or cost-effective. Hopefully my guesses weren't too way off. I like to think I've at least learned some things about parasites in the past few years. A book would certainly help though. Best wishes, Eric.
Okay.
I like to hear his thoughts, right?
I do like that. I like the working it through, thinking through the differential. So thank you for that, Eric. Marcus writes, ova and parasite to confirm if possible. As for the differential, most bacterial and viral agents can be excluded because of the 3-week history and lack of a fever. Might still be Campylobacter, though you'd expect a fever, cyclosporiasis, or Geodiasis, though the diarrhea should be more watery for both of these. Treat with 3 days of albendazole plus a broad-spectrum antibiotic for possible bacterial causes or superinfection. If confirmation is unfeasible, should also consider iron supplements.
Sue Ellen writes, hello again, TWiP hosts. This was such a tough case, but I came up with two possible parasitic diagnoses. There's also one bacterial possibility, shigellosis, but I'm going to set that aside since we do focus on parasitic diseases here. My two candidates are Balantidium coli and Amoeba histolytica. Both have several factors in common. One, both can cause dysentery. Two, both are common in areas with poor sanitation. Three, both are spread by fecal-oral transmission. While the bloody stools are not by themselves much to go on, I do lean more toward E. histolytica simply because it appears to have few symptoms other than dysentery, while B. coli usually is accompanied by fever, nausea, and vomiting, as well as bloody stools. One good thing is that metronidazole is listed as a treatment for both diseases, so absent a microscope to use to tell which parasite has infected our patient, it's possible he can be cured with metronidazole. I hope I got this one right.
Alistair writes, Dear TWiP, having missed the chance to send in my diagnosis for the previous episode due to being on holiday in West Africa at the time, which was, however, extremely interesting, especially as I got the opportunity to visit a diagnostic lab in Gabu, Guinea-Bissau, and see how diagnosis is carried out in such a resource-limited setting with the hospital lacking even electricity. Having guessed correctly, I thought I would try again this time and hopefully do better than my first attempt. I'll keep it relatively brief as this may be something of a shot in the dark, but I'm going to go for Amoeba histolytica. The symptoms of blood mixed in with partially formed stool immediately made me think of this particular infection, coupled with the fact that the patient is working in the fields and the fact that Amoeba are often found in the soil in the tropics. Finally, the lack of any fever, vomiting, etc. leads me to preclude any bacterial infections likely to have been the cause of this bloody diarrhea. Furthermore, as far as I am aware, Shigella and similar infections usually present as more acute and severe. As for treatment, I would prescribe a course of nitroimidazole, which Google recommends as very effective for the treatment of this condition. And in my professional opinion, I am inclined to agree. I am also attaching a paper that I have been reading at the moment for ideas on multiplexing for LAMP, which I mentioned in my last email, which you may find interesting, for distinguishing between pathogenic and non-pathogenic Amoeba species, in which I am planning to adapt the methodology for my own work on DNV, dengue virus. So you're getting a little virology here, which I mentioned in a previous email. I also wanted to add that you will be pleased to know, having said in a previous show when I wrote in before, that I will be transferring over to parasitology for my PhD at the University of Glasgow, working on a veterinary Helminth point-of-care LAMP assay. And as such, will probably keep trying to push this diagnostic test to you. And with combination with lyophilized reagents and lateral flow dipsticks, it effectively eliminates the need for cold chain and any equipment other than a heat block. There are also heat blocks which you can connect to smartphones, making this a really effective potential point-of-care test for infectious diseases in the resource-limited environments. I've attached some links below for a few papers on the subject. I've very much enjoyed your show so far and look forward to the next episode in case, and hopefully the chance to win a book one of these days. Alistair Antonopoulos.
So one of the papers Alistair sent is published in Scientific Reports, a smartphone-based diagnostic platform for rapid detection of Zika, chikungunya, and dengue viruses. How about that?
I like that.
It's very cool.
I was going to say to Alistair, so as I mentioned before, Christina Nalla at the one of the people that organizes the University of Glasgow's Global Health course has invited me. I'm actually going to be in January, the week of the 21st, over there teaching.
Nice.
So I will potentially meet Alistair.
Meet all these people, yeah. You should, meet Alistair for sure. Alistair, search out Daniel. That's next January?
January 21st in the afternoon. I think I'm lecturing that day.
All right. Kevin writes, Chicago weather, dull gray skies, equivocating drivel, 17 Celsius, unseasonably cool. This case calls to mind the old saw, Ars longus vita brevis by Hippocrates. TWiP, case 152, with its scanty clinical history, resulted in a lot of digging in the R's of medicine and threatened to brevis my vita. Defining the diarrhea. 3-week duration, we can classify it as persistent. This helps narrow the diagnostic possibilities. Acute is less than 14 days, persistent 14 to 29 days, chronic over 30 days. Number 2, defining the anatomy. The bloody diarrhea focuses attention on the large intestine. Small intestinal diarrhea has more malabsorptive symptoms with watery and or bulky stool. Colorectal diarrhea is characterized by the presence of pus, mucus, and blood, what is termed dysentery. 3, etiologies: infectious, viral, and HIV/AIDS, bacterial and mycobacterial, protozoal, helminthic, fungal, rare and often invoked by fringe health people, non-infectious, inflammatory, toxins, malignant. A persistent large bowel bloody diarrhea effectively rules out the common viral diarrhea such as norovirus and rotavirus. Duration of the illness makes enteric bacterial pathogens such as Yersinia campi, EA and ET, E. coli,
enteroadherent and enterotoxigenic.
Enterotoxigenic E. coli, Plesiomonas, Aeromonas, and Salmonella less likely, though not entirely impossible. The clinical scenario does not favor Clostridium difficile as a likely agent. Protozoal diarrhea from Giardia isospora, cytoisospora, enterocytozoan, cyclospora, and cryptosporidium usually do not cause dysentery. Helminthic disease resulting in dysentery is usually ascribed to trichuriosis. In textbook accounts, schistosomiasis has been reported to cause bloody diarrhea, but is not endemic in Panama.
Can I correct that and say cystoispora? Because that is my...
Cystoisospora?
Yeah.
Yeah.
I always do that. That's terrible.
The differential diagnosis in our patient can be narrowed to the classic infectious causes of dysentery in low-resource tropical countries, to wit, Amoeba histolytica and Shigella species, Dysenteriae sanae flexneri. Many people who carry E. histolytica remain asymptomatic, which could explain why the patient's family remain unaffected. The mind should remain open, however, to other background conditions such as TB, HIV, AIDS, though the patient's preserved weight and overall condition make these two less likely. Should also remember to consider folk remedy ingestion, cathartics, and purges, some of which contain mercury toxin exposure, agricultural chemicals, and inflammatory bowel disease, which is reported to be increasing in the tropics. Patient's remote location precludes most diagnostic workups such as nucleic acid amplification and antigen detection in the case of E. histolytica, stool culture for causes of bacillary dysentery. Field microscopy for accurate E. histolytica diagnosis is probably not practicable and may identify non-pathogenic Amoeba species. An empiric trial with metronidazole for E. histolytica and a quinolone or azithromycin for bacillary dysentery, if successful, would give a modicum of diagnostic certainty. If I must choose one diagnosis, I will say E. histolytica, though my brief look at Central American prevalence data does not inspire confidence. I close with a remedy from Pliny the Elder, died 79 AD, from chapter 19 of the Natural History. Dysentery is removed also by taking a spotted lizard from beyond seas, boiled down till the skin only is left, the head, feet, and intestines being first removed. A couple of snails also and an egg are beaten up, shells and all, in both cases, and made lukewarm in a new vessel with some salt, 3 cyathi of water and 2 cyathi of raisin wine or date juice, the decoction being taken in drink. Thanking you all for the ongoing excellent educational experience. And then Kevin gives us the references that have been consulted, which include a variety of papers in the CDC, and we will have those in the show notes if you'd like to look at them. I like this one, a survey of intestinal parasites, including associated risk factors in humans in Panama. It's from...
Yeah, this is actually a pretty good list of references here.
Yeah. Okay, you are next.
So this is...
Cullen.
Cullen?
Cullen.
Cullen. Hey, TWiP peeps. Okay. I believe the case of the man with bloody stools is caused by Amoeba histolytica. The blood in the stool is caused by the infiltration of the parasite into the intestinal wall. And because of the water sanitation, I think this is very plausible. I hope you guys are having a blast at ASM Microbe. Best, Cullen Lilly, microbiology technician, Atlanta, Georgia.
So I was the only one at ASM Microbe, Cullen. I enjoyed it very much. Did a lot of interviewing. I did an interview with the Amoeba Sisters. Do you know who the Amoeba Sisters are?
I don't, actually.
They're 2 women from Texas who have found success in a YouTube channel where they make very cute videos, mostly for grades 5 to 12.
Okay.
And they draw, and they use their drawings as part of it. And they used to be school teachers, and they were able to do so well on YouTube that they quit their day job, and they're supported by that.
That's great.
So I was interviewed along with them. And then I did a TWiV and 3 TWiMs.
Oh, wow.
So I was busy. And did you go, Cullen? Because you're right there in Georgia. I would think as a microbiology tech, you should go, because there's a lot of tech stuff at ASM Microbe. Peter writes, Bonjour, Professors TWiP. Just when you had all mastered the sweet mellifluous language of the Gael, I have migrated from Ireland to Montpellier, France, to work with my collaborators here. Before I left, I met with lab mate Gwen, visiting researcher Manon, and we invited Rosa Hughes, a high school student on work experience from Scotland, to join us as we listened to the last case. Due to the dysentery, we believe the man to be infected with the parasite Amoeba histolytica. Confirmation by stool examination. We also considered hookworm and strongyloides, but believe the persistent bloody diarrhea is more in keeping with E. histolytica. As non-eukaryote potential causes were also asked for, and I am here in the south of France, availing of their 16S metagenomics expertise. An honorary mention to Campylobacter jejuni and Helicobacter pylori as other potential causes of bloody diarrhea, although I must admit being very much a novice in this area and not in the possession of the microbial equivalent of parasitic diseases, 6th edition. A bientôt, Peter Stewart at TCD Parasitology. There is no equivalent of PD6 because it is the only textbook given away as a PDF free of charge.
I think that's true. I think
Sorry,
we were sort of landmark in doing that. I'm a little worried. So does this mean that the TCD parasitologists are going to stop listening to TWiP?
We can let them answer that,
I don't
right?
know. You know what they could do? It made me think of Microsoft. There was a famous guy, Bill Gates, I think his name was. And he and his wife were apart for a period of time.
Mm-hmm.
And they would go at the same time and watch a movie and then afterwards discuss it on the phone. And so I was thinking what the TCD parasitologists can do is, even though they're in different places, they can listen to TWiP at the same time.
For sure.
And then they could all Skype together and discuss the case.
Yeah, I think it must be fun listening to at least the case parts with a bunch of other people if you want to figure it out together, right?
I think that would be fun,
cool.
Going through the differential.
All right, we have one more from David P.
David P. writes, Dear hosts, I believe the man from the same Panamanian village featured in the past few episodes of TWiP is suffering from amoebic dysentery caused by Entamoeba histolytica. A listener from last week suggested the woman suffering from Geodiasis may have had E. histolytica, and the host mentioned that this may have been a reasonable diagnosis if there was blood in the feces, which there was not. This week, however, there is a new patient with bloody diarrhea, and E. histolytica can similarly be spread through rainwater, much like Giardia. The lack of external symptoms makes amoebic dysentery an easy culprit, and I am going with my gut.
Mm-hmm.
Thank you once again for the informative and entertaining podcast. Sincerely, David P. Okay.
I must say that I would also like to suggest E. histolytica, and these bacterial possibilities are interesting, but severity doesn't seem to be at the level of some of the ones that were suggested.
I think those. So I will go ahead and say that we, without any further diagnostic testing, came to a consensus that this was Entamoeba histolytica, and why? Why were we thinking that? And so those were the two main compelling things. You can go through causes of diarrhea. Think about viruses, which we, in general, some of them have vomiting, maybe watery diarrhea, but you usually don't think of viruses as causing two things: a bloody diarrhea, so a dysentery, or this degree of chronicity, right? Unless we're talking about a particular virus like HIV, and then it's sort of a unique setting there. When you go through the bacterial causes, and some people did a pretty good idea of listing them. Let's see, I think we had Shigella, Salmonella, Campylobacter, Shigellosis.
Right.
I mean, so they did a good job. The different E. coli's,
right.
Enterohemorrhagic, I think I would add. I don't know.
Would you expect the various pathogenic E. coli's to be transmitted in this setting?
Oh, definitely.
Yeah.
Yeah. I mean, in a lot of places, the short-born diarrhea, a big chunk of them can be E. coli. It's interesting that in a lot of areas of the world that Campy and Shigellosis are becoming the most common. So I would definitely have the E. coli in the list. Maybe not as common as we used to think to. And I know a lot of people have criticized earlier studies and the methodologies where E. coli was the number one cause of travelers' diarrhea. And now we're starting to question if that's really, well, probably not true now. And how true was it before? Or was it an artifact of the approaches and the methodologies?
Yeah, right.
So rethinking. And then we get into so I would say, you can get bloody diarrhea with a lot of those bacterial pathogens I mentioned. You often are sicker. You often have more prominent fever. And the chronicity, again, there can be an overlap, right? And that's why we love to do diagnostic tests. But in a setting like this, it wouldn't be characteristic for it to go on for more than 3 weeks, so to get into this level of duration. Then we got our different parasites, right? Giardia was mentioned. Cryptosporidium, Entamoeba, and B. coli. And one of the distinctions there, really the Entamoeba and the Ballantinium coli is where you start seeing dysentery.
Yeah.
And it's not clear that you can clinically distinguish those two. And not really having, as mentioned with one of our emailers, great prevalence studies, you sort of wonder how much of what is being attributed to E. histolytica might be B. coli. And I don't think we know certainly in these remote villages. When I've looked at some of the prevalence studies of diseases in Panama, a lot of the academics are in Panama City. And so it's hard to know, what's going on in the Panama City region, how much that corresponds to what we're seeing up in these remote islands by the Costa Rican border.
Might not, yeah. So what was treatment?
So treatment was good old flagyl.
Mm-hmm.
There wasn't really an issue with asking him to not drink for the week. So.
Mm-hmm. And did you follow up?
So he will actually, my wife was reminding me that right about now is when the group is going back to this area. So I need to shoot them an email and get follow up on the last few cases and see how everyone did. Because that's the, when you have resource limitations as far as your diagnostics, it's really great to try to get some feedback on, did this work?
Sure.
And they keep, Floating Doctors keeps good charts. So you actually can go back and see, they'll go back and they'll see this gentleman perhaps, and they'll say, oh, Dr. Griffin thought this and gave him a week of metronidazole, and what happened? So maybe they'll write in. They'll go through the cases. Dr. Griffin got this one wrong. This guy's still...
Now, I like this discussion on the anatomy, which was Kevin, right? Defining the anatomy. Bloody diarrhea focuses attention on the large intestine. So is that right? All dysentery originates in the large intestine?
So yes. And actually, the way Kevin did this, I thought was nice. I mean, there's always several ways to break down things. I always joke once that one is alphabetically, which is completely useless in medicine. But so the diarrhea duration, he did a good job. We have this often acute, or I should say very acute sometimes, right? Some of our viral can be.
Yeah. The noros are a couple of days, right?
Yeah, very acute. And then we have our, acute defined less than 14 days, our persistent. We get into this chronic where we're out 2-3 weeks, sort of past 30 days. And then he talked about the different anatomy. So if you're getting bleeding from inflammation in the small intestine, you can actually start to get some digestion of that blood. So you're not going to get bright red blood. The only way you get bright red blood from an intestinal source is when it is so brisk that the person is bleeding out from a peptic ulcer. And that's not going to be 3 weeks. That's going to be, an emergency. The blood in the colon, and I try to even break this down further. When it's mixed in the stool, it's going to be in the colon. If it's on the outside of the stool, then you think rectal.
Yeah, right.
So something maybe like even a bleeding hemorrhoid will give you on the surface. So you want to make that distinction.
So Amoeba does not reproduce in the small intestine?
The pathology is really in the colon. It's going to have these invasive, and some of the pathology is described as flask-like ulcers. Okay. And so that invasion is going to cause, interesting, you won't always see blood in the stool, but if you guaiac the stool, so doing a test for the blood, that'll be positive. But sometimes if you look for leukocytes, white cells, you don't see them. And the explanation has been that the amoeba are eating the white cells.
Got it.
And so they're not actually coming out, but the red cells are leaking out.
And what about the bacterial causes? Are they all focused in the colon as well?
No, not necessarily. So particularly Campylobacter and shigellosis would be characteristic small intestinal infections. Giardia, right, to throw up protozoan, also small intestinal.
And salmonellosis?
Also a lot of small intestine. So,
what about trichoriosis?
That's going to be large intestine.
Yeah. Okay. By the way, ars longa vita brevis. Art is long, life is short. And Hippocrates, of course, was the father of medicine, right?
That's what they say.
That's what they say. All right, we had 8 guesses. I left out David P. because he has now won twice. So
sorry to leave you out.
We'll leave him out. So let's do a random number generator for 8 again.
So while you're running that, I'll just make sure we do very simple life cycle here, right? I mean, you're basically ingesting the cyst stage, and you're getting the amoebic infection, and you're shedding the cysts and sharing them with your neighbor.
Number 7, which is Cullen in Atlanta, Georgia. How about that?
All right.
Where I just came from yesterday. So Cullen, send me.
You could have brought one with you.
Could have. Send me your address, Cullen. Send it to TWiP at microbe.tv. And thanks, everyone, for your entries. And there'll be another one shortly, so stay tuned. Now we have a paper for you, not only for your education, but your entertainment. Because our delivery is always entertaining, right, Daniel?
I sure hope so. This is a great article.
So this follows up on some recent things that we have done on TWiP. The title of this is Gut Microbes Egested During Bites of Infected Sandflies Augment Severity of Leishmaniasis via Inflammasome-Derived IL-1 beta. And this comes from the FDA, the Food and Drug Administration of the U.S., the NIH, and Johns Hopkins Bloomberg School of Public Health. The first author is Ranadir Day, and the last author is Shadan Kamawi. And so this is about infected sandflies spitting out microbes when they bite. And previously on TWiP 127, we talked about dependence of Leishmania survival on the gut microbiome of the sandfly. Connected. And another one, TWiP 137, role of a transmissible dysbiotic skin microbiome in inflammation during cutaneous leishmaniasis. So now our third paper dealing with the severity of Leishmania infection and microbiome. So there's a little arc here, no doubt, right?
That is true. We are building.
So what did you like about this paper, Daniel?
Well, the first thing I'll start with is I like papers that are published in Cell Hosted Microbe. I like the way the journal has a sort of a cover intro where they have a graphical abstract where they give you sort of a figure of what's going on. They also do a bullet point highlights where they say, this is what it's. So let me go through because that was interesting. So the highlights that they tell us are one, that as I think almost the title tells us, that gut microbes egested by Leishmania infected sandflies prime the host inflammasome. Then this inflammasome-derived IL-1 beta sustains recruitment of neutrophils to the site. And then they point out that if you give antibiotics to sandflies before transmission, you abolish this neutrophil infiltration. And then, concluding with abolishing neutrophil inflammation at bite sites impairs Leishmania dissemination. So they give us this really nice highlight. So we go into it saying, okay, I see the points that the authors are going to be describing. And then we get to walk ourselves through some really interesting biology.
Let me give a little background on the inflammasome. How about that?
I actually made some notes.
So you have some notes?
Think this is you go, because I think that as I realize as you go through, there's a lot of background you need to know.
Yeah, and I had to. So we have a nice description in our textbook, Principles of Virology, which unfortunately is not free, but the knowledge I will give you now from it is free here on TWiP.
Okay.
So the inflammasome, there are multiple inflammasomes, and these are protein complexes in the cell that are involved in stimulating inflammation. And the best characterized inflammasome is called NLRP3, and that's because it has it's composed of three proteins. The NLRP3 protein, which stands for NOCT, LRR, and PYD domain containing protein 3, an adapter called ASC, which stands for apoptosis-associated speck-like protein containing a card domain. You couldn't make this stuff up, right? And procaspase-1. The activation, so this sits in the cytoplasm. It's not active, but it requires two signals. And what it does, it releases caspase. So in the inflammasome, there's procaspase, which is an enzyme which is not active. It can't cleave anything yet. And with two signals coming into this inflammasome, caspase will come out active, and then that will cleave the precursor to IL-1 beta, becomes IL-1 beta, and that will then be secreted and then have its activities in stimulating inflammation. And the two signals are the production of signal one is the cell sensing some kind of foreign signal, like foreign nucleic acid or foreign
So our
protein.
DAMPs or our PAMPs
exactly
are danger-associated or pattern recognition receptors.
And the activation of NF-kappa B goes in the nucleus, and one of the genes that's transcribed encodes the precursor for IL-1 beta, which is the substrate for caspase. And second signal is typically toxins coming into cells or potassium efflux, reactive oxygen species, lysosomal damage, things that can be associated with bacterial infections. So that's another signal that goes into the inflammasome and results in the activation. So these two things together, you have pro-IL-1 beta, caspase, the result is IL-1 beta, which then stimulates inflammation. And that's why a focus of this paper is IL-1 beta, right?
Yeah. And actually, all you said, all these things are going to come up. They're going to be looking at ASC and IL-1 beta and NLRP3, right? This well-described inflammasome complex.
So my understanding, Daniel, is the sandfly is going to bite you, and the sandfly contains the metacyclic form of leishmania.
Exactly, which is the infective form. And I think we've talked about that before, right?
Mm-hmm. And this biting, and this is an interesting part of the paper, is, these sandflies will bite you and cause damage, which will cause inflammation, right? It'll trigger all the wound healing. It pulls neutrophils in. But when they're infected, and this is a whole thing in itself, which I find fascinating, they bite you even more, right?
Yeah, I thought that was interesting, right? Like that comes up in the discussion. Not only when they're infected and they have the bacteria as well as the protozoan in them, parasite, not only do they bite, but they bite you, they probe longer, they spend more time there, more local destruction. Really interesting.
So I'm really interested in what causes that. What parasite gene products cause that? Because the fly is not changing its behavior, right? It's just always a fly. It's just,
but what is it? What is the parasite? Because the parasite is changing the behavior of a sandfly.
Right. So this biting makes tissue damage, and that brings in neutrophils, and then neutrophils make things that cause inflammation. But if you just had a fly bite, that would go away quickly.
And that's the interesting, right?
The point here is that when the flies are delivering the metacyclics, the inflammation persists, right?
Yeah.
And in some cases, it persists long enough for the parasites to go away from the skin into the visceral organs, like the spleen, right?
Yes.
And that's what they're trying to understand here. Why is this neutrophil recruitment sustained when you have a parasite-infected fly as opposed to an uninfected fly? And there's
and I was going to say that actually brings us into, so now we've got our inflammasome understanding, but then we've also got to think about the particular leishmaniasis they're studying here. So we'll remind our listeners, there's the three main types, right? There's the cutaneous, which we don't worry quite that much about unless it's, let's say, on your face, where it can be disfiguring, where the parasites are replicating just locally.
Right.
And then the two other, we'll say, metastatic forms, the mucocutaneous, where it spreads all throughout. And then later on, we see disease at mucocutaneous junctions, or the visceral, which is actually what they're looking at specifically here, where it can spread to the spleen, as we used to say, reticuloendothelial organs, and then replicate and cause disease there. Right.
And as Daniel indicated earlier when he read those bullet points, what they conclude is that when the sandflies bite you, outcome, and this is the egestion, right? The opposite of ingestion, outcome microbes, which trigger the inflammasome, and that results in the production of IL-1 beta. And so you get this amplification by neutrophils. And it's the bacteria there. But as we'll see, it's not just them. They're needed, but they're not enough.
The neutrophils are bringing in, this is when the hunter becomes the hunted, right? They're going to be bringing in these macrophages and dendritic cells, but they talk mainly here about macrophages. And when those macrophages come in, they themselves become a niche for the leishmaniasis. Not only a niche, but a vehicle for the leishmaniasis parasites to then metastasize to these different organs.
So the microbes are essential for that. Without them, it will not metastasize.
Which is amazing, right?
But it's not enough to get to the spleen.
Yep.
Right. It's amazing, yes, that the parasite has evolved to take advantage of this. Those parasites that are doing this were selected for in flies that have this microbiome. And it's really interesting, but it shouldn't be surprising anymore because everything is polymicrobial, right?
But we also see, as we'll get into, the parasites are actually going to select the microbiome of the sandfly. They're going to modify it, and then we're going to see a certain almost marker. Well, it's a marker bacteria that we're seeing in the microbiome of a sandfly with a leishmania parasitism.
So let's look at the science here. So the first thing is the model. This is a mouse model where they apply infected sandflies onto the ears of the mice.
I thought this was great. I mean, they have the choice. Either they're going to inject them intradermally or they're going to let the sandfly do the application.
You compare the two, right?
And I think this is great because we often think, we got a model, we'll just inject them in there. And they show here that if your model was to sterilely inject them intradermally, you're not going to get success.
No, I mean, we now know that this is the case when any microbe is delivered by a vector. More things than just the microbe come. The saliva has various things, and for viruses, the saliva stimulates viral replication often.
Yeah.
And here we have the bacteria come in. So if you allow the flies to bite the mouse ear, you get dissemination from the skin to the spleen in about 5 weeks.
Mm-hmm.
And as you said, if you inject the metacyclics intradermally, it does not go to the spleen.
Yeah.
That's a cool result, right?
I think that is really.
So that's the bottom line here. What's causing that? And that's what the rest of the paper is trying to sort out.
Mm-hmm.
So they look at the innate immune response, what's going on in these ears. They do flow cytometry, and they look at neutrophils and monocytes from the ear. And they show that 48 hours after you either allow an infected or uninfected sandfly to bite, you get neutrophil recruitment.
Mm-hmm.
And that is, here these abbreviations, LG or LI, what do they stand for? LG is salivary gland sonicate, or LI is intradermal infection. You don't get anywhere near as much neutrophil recruitment.
Yeah, because it's key. They want to show, because other people have shown before, that the salivary components play a certain role in this. And they want to show that critical is really the microbes.
Now, here's one thing I wanted to ask you about, Daniel. They say another striking difference was an increase in the number of degranulated mast cells 3 hours after the infected flies, as opposed to all the other treatments.
Yeah.
Tell us, what is a degranulated mast cell and what causes it?
Yeah, so the mast cells are cells that have preformed granules sitting there. And people are probably familiar with these from the allergies, right? They get exposed to something and you have all this rapid response. You get hives, right? You get runny nose, things like that. And part of the very immediate immune response to a lot of things is mast cells. We've talked before. There's a couple of different ways. A lot of times this can be antibody mediated with cross-linking that causes it. Right. And that's kind of the most classic. But you also can have basically a danger-associated rapid degranulation. That's probably what we're seeing here. So
the parasite or something is coming in. It could be the microbiome, right?
It could. Well, I think what they're saying here, if you look at the graph, is it's the microbiome. It's all these bacteria. That is the strongest trigger, particularly in the first 3 hours, right? The most immediate.
And these components of the degranulated mast cells are things that promote innate responses. They lead to the production of two molecules, CXCL1 and matrix metalloprotease 9, which are recruiters of neutrophils and monocytes, right?
And actually, I thought, so this little nomenclature Bell was saying, I thought was really interesting. I mean, we talk about CCL and CXCL, and right, so the CCLs, it just is sort of a structure of two cysteines, and then X is there's something between the two cysteines in the structure. But more important, the MMP9 is this matrix metallopeptidase, and it allows neutrophils to cross basement membranes. Because normally, right, you're going to have this barrier, and the neutrophils are not. So this is not only going to attract them, but it's going to create pathways for the cellular recruitment. So I thought that was really fascinating.
So that's where the matrix in the name comes from, because it digests the matrix, and that's right. It makes it permeable. Yeah, that's pretty cool. So they looked at transcription of these two chemokines, CXCL1 and CCL2, and also MMP. They're all induced after the bites of the fly, infected fly, compared with all the other treatments.
And when they do like their heat map, right, it's the only time that you're seeing, for instance, MMP9 is when you're actually letting the natural, fly bite with the microbes, everything.
Right. So saliva alone is not enough. Parasites injected alone, it's just the fly biting is delivering them.
Yeah, which I thought was really, it's showing how critical, as we'll later see. Should you get rid of those microbes, what will happen?
So MMP9 will recruit the neutrophils and, as you say, let them go in. They say it orchestrates a positive feedback loop, which means more will come in then, because it's recruiting them, right? Once it's produced, it will recruit them in. They also see a lot of induction of IL-1 beta, which we just talked about as being
the end of that cascade.
The cascade from the inflammasome. And so this happens in the infected and uninfected fly bites. IL-1 beta goes up, which they say maybe this activates the inflammasome. So they then look at markers of the inflammasome, right? They not only IL-1 beta, which is a product, and they do look at IL-1 beta, and this they find really high after infected flies compared to uninfected flies, right? So both can induce it, but much higher with the protein itself is induced at much higher levels when you use a fly to deliver the parasites. Exactly. And they find that neutrophils are the source of this IL-1 beta.
Yeah, and I think that's the sort of early point they're making here is a lot of what they're trying to show, I think, is that the inflammasome activation is not brought on necessarily by the parasite, but by the microbiome that's being injected.
Right. And that's where the feedback loop comes from, because initially the neutrophils are brought in, and then they produce IL-1 beta, which bring in more neutrophils.
Exactly.
Right. And then they ask, is the inflammasome activated? They look at this ASC protein with the funny name, the speckle.
Yeah, so now we're moving on to figure 2.
And they show that these proteins are up, the inflammasome is there, and it's assembled. So that is activated as well. And they say that this is nothing new. They have been, leishmania is known to activate this NLRP3 inflammasome. So what they do is they take bone marrow derived macrophages. They incubate them first with lipopolysaccharide, and then they add L. donovani parasites. And you get a lot of IL-1 beta mature, in other words, cleaved, only when you add LPS.
Yeah.
And not just parasites. So that's, I think, a clue that the microbiome is involved.
I thought that,
right?
Well, it's pretty critical.
Yeah, it's totally, I mean, they say
no LPS, nothing. And
they say if you add a ton of parasites, you can, you can get more IL-1 beta, but, maybe there's not a ton coming out of a sandfly, right? So LPS is improved.
And the LPSs are classic, just for the listeners, is your classic gram-negative surface stimulant of our danger.
That's right. So then they say, okay, the midgut microbes of this fly must be doing something here. So we're going to look at this. And they do a cool experiment where they take wild-caught sandflies and they let them bite an agar plate, basically.
Yes.
Right? They have a warmed LB agar plate.
Yeah.
And they put the fly in it. And the flies think it's skin, I guess, and they bite it.
Yes.
And then they take the flies out and they put it in the incubator, and they can see colonies of bacteria growing wherever a fly bit it, right?
Yeah.
This is great.
Yeah. It's really creative.
So then they can take the colony, extract DNA, and look for leishmania and see which one came from a foot of a fly and is just a contaminant versus injected that has leishmania DNA in it. Right? And then they say, well, what are the bacteria that are in here?
I'm trying to think of who was like the graduate student who was doing this, and his mentor was like, "What are you doing? What? Oh, they're biting the agar. I made it warm."
That's great. That's really
it's great stuff.
So here are the genera, and I have to say, I don't know a couple of them: Sucamurella, Lysinibacillus, Pennibacillus, Solibacillus, and Bacillus. These are all found in these.
So I'll try to break this down real. So you probably are listeners who cross-fertilize themselves, maybe have gotten a little bit of the focus on the different major phylums of bacteria that we're seeing in the gut.
Right.
And the two really big ones that we often talk about are the Firmicutes and the Bacteroidetes.
Right.
And there's sort of the idea that one group is a little bit better than the other group, and one is associated with, we'll say, developing world populations with parasites. And then when you change that, they shift over to the obesogenic. So this sort of balance between the two major players. But also here they're bringing up a minor player that we don't really talk that much about in the gut microbiome literature, which is this Acinetobacteriae. And that's where they actually find that signature bacteria. So the Zucca
Yeah, I said it's a signature for infected midguts.
Yeah. It's interesting.
Go back to what you were saying earlier, that somehow this is selected for in parasite-infected flies. So I wish we had Dixon here to tell us about these flies a little more, and
yes,
how long they're infected, if it's their whole life, right? Or if just after a blood meal, how often they take a blood meal. I don't know any of those things.
Yeah. I mean, they become infected after a blood meal, but then, well, where's Dixon when you need him?
Yeah, right.
In China.
All right, so now what would you do? Well, if you said, I'd feed the flies antibiotics, you'd be right. Feed them antibiotic cocktails to try and deplete the microbiome and see what happens on, in the mouse ear. See if this inflammation still happens.
Yeah. So
they do that. They show that the microbiome is decreased. It's not eliminated, okay? It's just decreased. And then they let these antibiotic-treated flies bite the mouse ears. And then they look at IL-1 beta. And it's down compared to flies without antibiotic treatment. NLRP3, procaspase-1, cleaved caspase, they're all reduced when you take away the microbiome or when you reduce the microbiome. So basically, these bacteria are important for the inflammatory response, the neutrophil-based, inflammasome-based inflammatory response when these flies deliver parasites to the ear of the mouse. Did you like that experiment?
I mean, I loved it. And I thought the final figures and the final subfigures, so, figure 4, where they said, okay, we treat them with antibiotics versus we didn't. And
they also had an antagonist there of IL-1, right?
Exactly.
Yeah.
And you basically, I thought this was great. If you leave them as is, you get dissemination to the spleen in the majority of cases.
So that's the other part. That's besides the reduced inflammation in the ear.
The ultimate,
the spleen doesn't get colonized after you use antibiotic-treated flies,
which is huge. Which I thought was really, because that's what we really care about in the strains that visceralize. Is we don't want them to go to the spleen. We don't want to see the cala-azar.
And they do an interesting reconstruction. They inject parasites with bacteria. This solely bacillus bacteria recovered from a bite on the plate.
Mm-hmm.
With or without LPS. And they can almost reproduce or partially reproduce the inflammation after injection by adding these bacteria, but it doesn't go to the spleen.
Yeah.
It does not go to the spleen. So you need microbes for inflammation. You do need them for getting to the spleen, but it's not enough. It's something else in the saliva, right?
Yeah. And we know that from other studies, right, that there are salivary components that are critical as well. So you need it all.
So I guess they could try fractionating the saliva and see, what when added. So this is the reconstituted system. You inject the parasites along with bacteria and then some salivary component. Mm-hmm. And you reproduce the metastasis to the liver. And if so, what is it? I'm sure they're trying to do that. So the microbiome is collaborating with the parasite in this case to allow the parasite to get to the spleen. Now, Daniel, why does the parasite have to get to the spleen?
That's an interesting question.
In other words, what's the function?
What is the advantage for the parasite, right, to get to the spleen? And I think the survival advantage, the evolutionary selection advantage, is this is its place where it can last for many, many years. I think we presented a case where it had been over a decade since the individual had been infected.
Yeah.
So rather than just a localized infection that might be self-limited, this is an area where people go on for years, and they can then be sources of other
and transmission
sources.
I mean, that's the key. Because if you went to the spleen and that was the end, there would never be any evolutionary selection for that.
So there has to be feeding into the transmission.
Because in the end, it's always about transmission to a new host, so you can make more parasites, right? Yeah. It's the same thing. That's why herpes latency in your peripheral ganglia reactivates periodically, so the virus can spread to another host. It wouldn't do any good if it stayed in you.
If it stayed, it
would be the end
dormant.
of it. Yeah, so that's very interesting. So here we have bacteria collaborating with parasites to, and I wonder, now the bacteria I'm not sure if this is true. You think the bacteria might be picked up by another sandfly at some point, or is that the end for them? Are they being used by the parasites?
Yeah. So how is the? How is the?
I have a sense that this is the end of the road for these bacteria that are delivered, and they just.
No, I think that's true. I think the bacteria that get egressed into the bite site. Yeah. They're eventually going to get killed by the immune system. So they're not going to propagate in any way.
Yep. They conclude by saying the significance of vector microbiota likely transcends sandflies and leishmaniasis with analogous ramifications for other vector-borne diseases.
Yes. And they mention the hot topic tick.
That's right. But yeah, ticks also... A variety of mosquitoes have been shown to deliver things with saliva. Let's see. Cruise neutrophils also. This is neutrophil recruitment in virus-infected mosquitoes. I'm not sure they've looked at microbiota, but they should.
Mm-hmm. Yeah, no, that's a nice thing, and they open up a lot of other avenues of investigation, and. In our
some
area,
ticks that transmit bacteria of medical importance. Yes, so they're talking about bacteria in the salivary gland of the tick there, and of course those are transmitted actually.
Mm-hmm. So interesting. I mean, they do make a point. This is sort of a part of the paper that's a little challenging. Is they bring up the fact that it's ultimately the macrophages. And they don't maybe do as much macrophage work as, in the main figures as you might like. I actually thought it was interesting that a few of the supplemental figures are looking at the M1, M2 paradigm and the kind of shifting and impact that would have. But that didn't really make it into the main paper.
No, they say in the second sentence of the abstract, a prominent feature of vector-transmitted leishmania is the persistence of neutrophils where they protect captured parasites, but we don't see any of that in this paper. Right?
Yeah, we don't.
Yeah. It'd be interesting to see. I mean, how do you think they protect them? They don't get inside them, right?
We think of them as being inside the macrophages and dendritic cells, so it's unclear to me what sort of how the mechanism of the protection is occurring. But interesting stuff. I like this paper.
Very good.
So I guess now we're going to just have to leave out baits for the sandflies that adjust their microbiota.
Well, that's an interesting question. Is that a way to control this, is to give antibiotics to the sandflies?
I mean, you sort of look at this when they did that, when they gave them antibiotics, they prevented, visceral leishmaniasis. And you don't have to make them sterile, potentially. It may just be you have to do something that modifies the microbiota. So we see in us, right? If we eat too much sugar and get rid of our parasites, that we have a obesogenic intestinal microbiota. So couldn't there be the planting of different plants that actually affects the parasites?
Here again, we need Dixon with his ecological knowledge.
Yeah, because this would be interesting. Like, are mangoes or figs, or what are you planting in these areas that might be critical for this particular bacteria?
My suggestion would be, how would this work? You tell me you get a sandfly bite and you immediately put a topical antibiotic. Would that help, you think?
It's interesting. Maybe. I mean, is it getting in deep enough?
Yeah.
Yeah, and that also raises the issue because it's a period of time that's occurring, as we're seeing. There's, if you got a dose of an antibiotics, or were taking an antibiotic when you got bitten, would it protect you from leishmaniasis?
Of course, we often don't catch it early enough, right?
Well, that was, so an interesting thing, right? Our troops, we normally think of certain types of leishmaniasis as having the ability to visceralize and others not.
Right.
But what we saw in our troops, the U.S. troops, when they were over in the Middle East, they would have visceralization of what normally were cutaneous forms. Uh-huh. And had they been on a daily prophylactic antibiotic of some sort, would that have affected? I don't know.
Yeah. What's the treatment for leishmaniasis?
It's for, this is the liposomal amphotericin, or the miltefosine, or the stibogluconates, or the pentavalent antimonial. So
amphotericin is
pretty
also
bad stuff.
Antibacterial, right?
Mainly an antifungal.
Fungal? Okay. Yeah. I'm wondering if you threw in an antibiotic cocktail, would that help? I think you have to catch it early.
But I guess
enough,
early,
right? It has
because
to be
once it's visceralized, it's...
So can you tell when you've had a sandfly bite? Is it a nasty bite?
No, they're actually, so as opposed to the tsetse fly, where Dixon always does his shifting hands, they're really like cutting you.
Yeah.
And it's interesting. We always say, oh, you know when you've been bitten, but then there's cases where people end up with sleeping sickness and like, I don't remember being bitten. So I'm not sure how true that is, but the sandfly is a beautiful, tiny, very delicate insect.
Yeah.
And you don't necessarily know you've been bitten.
So I think the topical antibiotic probably wouldn't help because people just don't know it, especially in endemic areas where they're bitten by all sorts of things.
But I do wonder about, so the sandflies, right, are living off, sugar diets like a lot of these insects. I wonder if there's a way of modifying what you plant in an area, because the sandflies, as I mentioned, they're really delicate. They can't go very far. Their range is limited. It's a few yards. They're not even great flyers, more get up about 2 feet. That's why a lot of the bites tend to be below the waist unless you're sitting on the ground, laying on the ground.
Got it. Well, if there's a miracle and Dixon listens to this episode, maybe he'll join.
Maybe he can come up with something in his vertical farm that will change the microbiome of sandflies to disallow their transmission of the parasite.
I mean, it sounds tough to me because there's so many plants out there. But if it's known what they feed on, if they preferentially feed, then you could put a lot of that out there and distract them. Maybe. Who knows?
Yeah. I mean, we talked on one episode about how the introduction of the mango to West Africa has increased the malarial burden because that actually increases the fecundity. And,
you plant marigolds in your garden to have what's the effect of marigolds? Marigolds I think it's on insects.
Yeah, it
They're
is.
like repellents of insects. Marigold is the most well-known plant for repelling insect. There you go.
Yeah. Is it a permethrin type effect? I'm not sure what the chemistry of it is.
Don't know.
But it is interesting, right? We go ahead and we're always, all these mass campaigns and all these, we're worried about resistance in the parasites. Can we do things where we're changing the natural environment in a way through different plants? And
have you another case for us, Daniel?
I do, actually. And I thought I was going to have fun with this case. I usually write them up. But I did, and I wrote this one up, of course. But I was going to sort of present it to you, and you and I are going to walk through it together.
All right.
So, this is a gentleman who comes to me in clinic. I saw him not too long ago. And I'll mention, maybe you'll be curious why he's coming to see me. But he says, but you know what, Doc, while I'm seeing you, I had this concern. I was in the city recently, and after coming back, I noticed that I had developed a rash down in my the area of my parts, down in my groin area, and I'm a little concerned about it, and hope that you could help me with this. So this is always the fun part as the clinician. So you get to start asking him questions. The first question was, so what were you doing in New York City? And so he says, well, you know what? I have an open relationship with my male partner.
Okay.
So I was in the city, exercising the freedom that comes with being in an open relationship. And so he was in the city and he had a sexual contact while in the city.
With another male, I presume.
With another male that he had met through. It's interesting. A lot of the connections now, they use a lot of social media apps to hook up. Mm-hmm. And so you sort of put on your alert. I'm a male and I'm interested. And so there's connections. And so he has this relationship. But what we realize is isn't someone he knows well. So he can't call this person back up and say, hey, I've got kind of this rash now, and I'm wondering if it had anything to do with our
how long after the encounter did this rash develop?
So he was a little vague. He wasn't quite sure because this was not the only time he'd been in the city to have one of these exposures. But in his head, he was connecting them. Maybe a couple weeks, he thinks. But then he had other encounters before that.
Was the rash on his penis or scrotum or on the skin in the groin area?
So it's actually not on the penis itself. It's not on the. It's in the area. It's in the pubic hair.
Pubic hair.
So in the pubic hair, and what he's concerned about, it is very itchy.
Okay. Yeah, that would be a concern, right?
Okay. And he is, just to give background, the reason he's seeing me is he sees me every 3 months for his PrEP for his pre-exposure prophylaxis for HIV. So he's HIV negative, and he's getting his pre-exposure prophylaxis.
At least he's doing that, right?
So he's doing that.
That's good.
Yes.
That's good.
That is good. So he examines the area, and he notices some things there. One of the things that he and I notice is there are little tiny sort of, rash is mostly around the umbilicus, so the belly button to the, and it's you can see that he's been scratching, and there are little spots, and I hate to use the word blue, but yes, there's little blue spots. It's a erythematous rash in this area. He's used some over-the-counter products, which he says helped a little bit, but this still hasn't resolved, and he's still concerned about this.
Blue spots.
Little blue spots as part of this rash.
Are they moving?
No. So the little blue spots are actually skin changes.
Okay.
But hopefully this is one that people will enjoy. So he actually, because he's concerned about it, he buys a magnifying glass so he can look a little bit closer. And he describes actually seeing things in this area, which we see as well on exam.
Okay.
So very small things that can be seen with a little bit of magnification.
Interesting.
But otherwise, past medical history, not much as far as surgery, not allergic to anything, not a smoker, not a drinker, but he is sexually active and open relationship, as we mentioned.
All right.
And that's the...
So what would you like our listeners to do to give you a little differential and then a little...
I think a few things would be, have a, what do you think this could be? Give us a little bit of a differential. I have to admit at this point, I was pretty much like, okay.
Right.
And I think he pretty much knew what was going on too. So not only do you want a differential in what people think is the diagnosis, but how do you approach this?
Right. Yeah. And you would like them to say, what would you do to confirm it, right?
Yeah, that would be also. Yeah. How do you confirm this? How do you make the diagnosis?
Okay. Very good.
What could be masquerading?
All right. There we go. TWiP 153. Should we wrap it up, Daniel?
So I think we could. We have a few emails, which at some point when Dixon is back, we can read through, but.
Yeah, I think we should do an episode with just a case and email. What do you think about that? We'll do one without a paper.
I think we could do next time, because I feel like we're getting a little backlog here on the emails.
Yeah, we have a lot of emails. But I'd like to have Dixon in with us to do that. So that's TWiP 153. You can find it at microbe.tv/TWiP. And of course, if you have a phone of any kind, you have a program on it. And if it's an iPhone, it's called Podcast. And you can just open up that program and search for TWiP, This Week in Parasitism. You might also get This Week in Photography, but, if you're looking for parasitism, find us, subscribe. You get every show automatically. It's free. You don't have to pay, and you get them all. And why not? Just subscribe and, listen to the ones you want. Why do I tell you to subscribe? Because it gives us numbers. We know how many people subscribe, and we'd like to know, that helps us to secure a little bit of income. To know that we have lots of subscribers. So please subscribe. And if you do like what we do a lot, you could consider becoming a patron, supporting us financially. Go to microbe.tv/contribute. We have a number of ways you could do that. And of course, send us your questions. Send us your case guesses to TWiP at microbe.tv. Daniel Griffin is here at Columbia University Medical Center. I guess it's Columbia Irving Medical Center.
It is.
Columbia University Irving Medical Center. CUI. I have to put an I in there. And he's also at parasiteswithoutborders.com. Thank you, Daniel.
Pleasure as always.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to Ronald Jenkins for his music and ASM for their support. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.